Hi, this research project is about investigating the neurotoxic effects of polystyrene microplastic. Neurotoxicity refers to any sort of disruption in the nervous system by foreign substances called neurotoxins. Neurotoxins are abundant and arise from various different sources. One such common neurotoxin is plastic, which is an abundant compound in the environment. In recent years, plastic production has increased significantly, reaching over 460 million tons per year. Concerningly, plastic are subject to fragmentation and degradation, which leads to the production of something called microplastics and nanoplastics. Microplastics are tiny particles measuring less than 5 micrometers in size, usually categorized between 1 to 5 micrometers in size. They can be divided into primary and secondary microplastics. Primaries are manufactured deliberately, such as pellets and microbeads, which are now present in food wrappers, consumables, and cleaning agents, whereas secondary microplastics are not intentionally created but result from the breakdown of larger plastic items. The breakdown is done through just merely exposing plastic to UV, which makes it a very big emerging concern. We end up inhaling these microplastics without knowing. Moreover, microplastics are known for their stability and inability to break down, making them an emerging pollutant globally. The most common microplastic being used in consumable goods and left outside in food packaging and cosmetics is polystyrene microplastic. MP exposure causes toxic effects on the gut, liver, brain, kidney, and other reproductive organs. In this study, the brain is the center of investigation. MP exposure has been linked to neurodegenerative diseases like Parkinson's and Alzheimer's, as well as other potential risks to human health. In this study, the toxic effects of increasing doses of polystyrene microplastic exposure were assessed in the human neuroblastoma cells by measuring cell viability using an XTT assay for doses ranges from 200 to 500 microgram per mil, and the downregulation and upregulation of two proficient genes was studied. Cell proliferation gene, PCNA, largely reflects on cell proliferation activity, and it's commonly used as a marker of cell proliferation in healthy and tumor cells. Examining PCNA expression in Shy-Y cells subjected to increasing doses of PSMP is crucial for understanding cellular response to these particles. And on the other hand, autophagy is essential for cellular homeostasis and degradation of damaged components, and it is influenced by polystyrene microplastics. Monitoring the expression of beclin, which is an associated gene of autophagy, provides insights to whether polystyrene microplastics induce or inhibit autophagy, thereby affecting cellular health and toxic mechanisms. To determine the type of plastic to be used for this experiment, two different types of plastics were validated. Polystyrene macroplastics beads were dissolved in toluene and then grinded with a mortar and pestle to form a fine powder. And DFPE polystyrene microplastic beads, which were purchased commercially, were found in clumps, and the individual particle size was determined by resuspending the powder in DI water and sonicating the suspension to create agitation, and it was measured on the citation 5 using a 30 micrometer scale. As it was evident, the DFPE microplastic polystyrene beads were exactly 5 micrometers big, and the polystyrene macro beads did not grind into a fine powder. So the optimal choice for this study was the DFPE polystyrene microplastic beads. Based off that, the treatment was prepared by sterilizing the microplastic beads through autoclave and resuspending it in sterile culture media. XTT assay was performed by testing 200 to 500 microgram per mil doses. The 400 microgram per mil dose was chosen to be the most optimal dose to move on with PCR. Although not significant, the downregulation of the PCNA cell proliferating gene was observed, and the upregulation of beclin was observed. Here is a visual representation of what the 400 microgram per mil dose treatment did after the cells were left with the treatment for 24 hours. In conclusion, despite encountering challenges during the experimental procedure, such as difficulties in the polystyrene microplastic preparation and PCR analysis, the study yielded important results indicating dose-dependent cytotoxic effects and alterations in gene expression levels. The hypothesis was tested and proven to an extent. The increasing doses and prolonged exposure of polystyrene microplastic did decrease cell viability by inducing autophagy and a decrease in cell proliferation by downregulating PCNA expression and upregulating Becklin expression, although not significantly. There are many future implications of this research. If it were to continue, I would suggest that more apoptotic and autophagy-related genes to be explored. There would have been significant results if done more meticulously. This research offers a glimmer of hope for this to be possibly explored. Another thing